Transcription part 1, DNA strands coding versus template. This is the and DNA, 3' and DNA, and 5' DNA, and there be prime RNA being made, and this is the direction of transcription. So from the 3', this is going to be 5' and 3', and this is the 3' end of the RNA, and we call that strand the template. DNA strand, and this strand the non-template. DNA strand. So for coding, just non-template. DNA and template. DNA and RNA. When the base pairs match, we'll see AUG, which is the start codon, and TAC is going to be the pair from the template DNA, and we'll see ATG, which T and U are different, distinct from RNA and DNA. And let's say we continuously have T, G, A, C, T, T here. Then we'll have A, C, U, G, A, and A, C, T, G, A, where that part and that part matches. That's why we call the non-template DNA the coding DNA, because the coding DNA has a similar sequence to the RNA that is being transcribed. Okay. Gene structure and transcription unit. So let's say there is a promoter for transcription, and next to promoter there will be a transcription start site, and we number it +1, and will be 5' UTR, intron 1, and exon 1, exon 2, intron 2, exon 3, that includes 3' UTR, and there will be a transcription termination, where at the downstream of the promoter, there will be, so this is going to be considered upstream, and this is going to be the downstream. Okay, so at the upstream region, there will be some regions that has upstream regulatory elements from the transcription start region to right before the termination is going to be considered the transcription unit. And from the upstream regulatory elements to the end of the transcription termination is considered a gene. So gene is a DNA segment that determine DNA segments that determine an expressible unit. Which result in the formation of a specific functional gene product. And this gene product can either be RNA or protein. Transcription unit is a contiguous stretch of gene that encodes the sequence of the primary transcript. So this includes coding regions, introns, 5' and 3' UTR. A promoter has a minimal sequence needed. Sequences needed to initiate correct transcription. Note that often use of the term promoter is meant to include upstream regulatory sequences as well. So sometimes this term promoter includes upstream regulatory sequences. Okay. RNA polymerase. RNA polymerase 1 catalyzes the formation of RNA. Two, uses one strand of DNA as a template. Three, RNA synthesis occurs. In the 5' to 3' direction, that if you see the picture one more time, the template DNA was read from 3' to 5' direction, and the formation was from the 5' to 3'. Only a protein, or only a portion of the genome is transcribed in each cell. And this makes cells different from each other. Only a portion of the genome is transcribed in each cell, and this makes the cells different from each other. So let's go to this picture one more time. So this picture shows the coding strand and the template strand, and RNA polymerase reached template strand to produce an RNA that is the same sequence as the coding strand. Good. Now then, let's talk about RNA polymerase enzymatic activities. It has a polymerase activity. So polymerase activity is that it synthesizes RNA nucleotides into RNA polymer. It has a helicase activity. That it melts the duplex DNA so that the template strand can be accessed. It has a dissociate activity of, or to dissociate the RNA from the DNA after synthesis. So during transcription, only 8 to 9 RNA nucleotides remain duplexed with the DNA at the moment. So we see that when the new RNA is forming, there are only 8 to 9 duplexed formed RNA that exists. Okay. So we talked about gene structure, that a gene is made up of the regulatory elements. They are the promoters, upstream regulatory regions, and transcription start site and transcription stop site. All right. Eukaryotic RNA polymerase II gene. This gene includes TATAs that we call Tata box, TATA element, RNA polymerase II binding site. And then it is usually located in -25 site. There will be UR E, the upstream regulatory element, and this is going to be the transcription start site +1, and after that there'll be 5' UTR, and this is going to be the exon 1, intron 1, exon 2, intron 2, exon 3, and exon 3 includes 3' UTR, and there be transcription termination, and an important A signal for transcript cleavage, and poly A addition. Nothing very specific on this picture.